start this out. Our next speaker is Fangue. She is currently at UC Davis, but she was previously doing a postdoc with Gretchen Hoffman. So she'll be talking to us about their use of genomics in ocean acidification research. Take it away, Nan.
Thank you.
You're welcome.
Yeah, so thank you for having me. It's a pleasure to be here. Gretchen is sad that she wasn't able to come, but she wasn't able to sort of manage it with all of her travel plans. But it's a pleasure for me to be here and be able to speak to you all. So thank you very much. And so just for a little bit of context, so what we're trying to do is gain ecological insight using genomic approaches. And so in general, animals are faced with some sort of challenge in their environment. And the way that they integrate these environmental signals is transduced through their genome to the whole animal, and then the whole animal then responds in some way and performs a response to this environmental signal. And those are the sorts of things that physiologists like me are generally interested in making measurements on. And then this differential performance then is transduced up into things that ecologists are usually interested in, so distribution, abundance, population dynamics, those sorts of measurements. And so what I want to talk to you today is the use of genomics as a tool for measuring differential performance. And so we're going to be focusing back here in the genome section. So for today's talk, I've sort of broken it into three different things. And so in the first part of the talk, I'm going to just sort of give a basic overview of what omics techniques are and what they have to offer. And for those of you that don't have a lot of molecular biology in your background, I'll just sort of walk through some of the techniques, certainly not the entire genomics field, but certainly the techniques that are most relevant for the types of questions that we're interested in answering. The second part of the talk will be a demonstration of how we can use transcriptomics, in this case, for ocean acidification research. And I'll show you some of the experiments that we've been doing in the Hoffman lab using the purple sea urchin. And then finally, I'll end with just a couple slides on next-generation sequencing technology. So this is some new technology that is coming to the field of molecular biology that we think has some relevant applications for ocean acidification research in the future. So ecological genomics. Well, genomics is simply the study of the structure and the function of the genome. And the ecological aspect is when you study the structure and the function of the genome with the aim to understand the relationship between the organism and its biotic and abiotic environment. So when we're talking about genomics, it sort of includes the structure and the function of the entire genome. And this is a really great resource if you get really interested in ecological genomics and you want to just start sort of reading generally about the field. This book doesn't have a lot of technical protocols and those sorts of things for actually performing the techniques, but what it does have is a lot of context for how you can apply these different genomic and molecular techniques to answer ecologically relevant questions. So it's quite a nice book. And in this book, they highlight how ecological genomics fits in all the different subdisciplines with which it draws upon, so genetics, evolution, microbiology, and then here, physiological ecology and physiology, which is where we'll be focusing our tour through ecological genomics today. And so omics has many different types. You hear all these words bouncing around, but simply genomics is generally focused on the gene, the DNA, the gene sequence, or the genetic code. Transcriptomics, which will be the focus of a lot of the data that I'll show you today, is the investigation at the level of RNA, or the actual messenger RNA that's derived from the code. You can have proteomics, which is the study of the proteins, the enzyme structural proteins that are translated from the message. And then finally, the study of metabolites is another subdiscipline called metabolomics. So that's sort of the breakdown of how of omics. And if you want to take a molecular approach to an ecophysiological question, this is a good way to sort of think about how you approach a study like this. So one thing that you want to do is you want to sort of decide what genes you want to study. So what genes are interesting in the particular context that you're doing your experiments in. And there are sort of steps along the way that you need to do. So you might need to clone the gene. So that's gain sequence information for that particular gene of interest. So learn the sequence. and you might want to do some characterization of the genes, so how they're organized in the genome, what their promoters regions look like, things like that. Another thing you can do is you can determine the mRNA expression level of those genes, so you can look at the transcriptome, so things that are being expressed in response to some particular stressor, for example. You can also study the protein products, proteomics, and you can examine how these genes are regulated, so what's controlling the switching on and the switching off, or the upregulation and the downregulation of these genes. And so for today, I'm going to primarily focus on transcriptomics. So transcriptomics is simply the study of the transcripts, or the RNA molecules, and generally what you're looking at is messenger RNA, so the things that will ultimately be translated into proteins. And so why you might measure mRNA expression, or why this measurement is important, is, as I said, mRNA is the template for translation into protein, which are the functional units, things that make the cells really go bing. The changes in mRNA levels often occur very quickly. So you give an animal an exposure to an ocean acidification stressor, for example, and you may see changes in the gene expression patterns within minutes. So you can see responses really, really quickly. You can use these types of measurements then to obtain a profile of gene expression for a given condition. So they will give you a hint at what genes are important under a certain subset of conditions. And it's a very sensitive physiological indicator, and we have very sensitive quantification technologies to make the measurement. So we really can see very small changes that you may not be able to see at the whole animal level with a more holistic measurement. We can see indexes, or we can use this as an index of change, a very sensitive indicator. And it's been very popular with ecophysiologists. Particularly, lots of people are using it in the marine community, which is good because we have lots of resources that we can apply to questions of ocean acidification. Okay, so I'm just going to kind of run through a hypothetical example about how you might use transcriptomics in an ocean acidification, for example. And so if you want to understand the mechanisms of the stress response, for example, so if you're deciding what genes you want to study, one way you can approach this question is you can take a candidate gene approach. So you can use your knowledge in this case of genes that are important in a stress response to pick some candidate genes to look at. And so that's what I'm going to describe how you might do that first. Okay, so in this example, if we're interested in the stress response, we know that heat shock proteins are involved in an animal's ability to deal with a stress response. So it's just a class of proteins that help when an animal is undergoing stress and they have some damage to their protein pool. These molecules come in and help to stabilize and repair some of the damaged proteins. So it's an indicator that an animal is undergoing stress. And so if you wanted to study this gene, then what you could do is, if you were lucky enough to have an animal or be studying an animal that had a fully sequenced genome, you could go in to the database and obtain the species-specific sequence for this particular gene. So look for heat shock protein sequence. Or if you don't have an animal that has a fully sequenced genome, you could go in and obtain related sequences from a database from other organisms. You could align these sequences, look for areas that are conserved, and then go through the process of designing the primers and cloning the genes. And so all that process does is it's a way for you to actually gain sequence for the particular gene and the species that you're interested in. And at that point then, once you have the sequence, you can develop gene-specific primers as a way that you can actually go in and probe your sample to assess how many transcripts of that gene are there. Okay, so once you have that information then, or I guess let me go here first. So in a recent paper from the Hoffman lab, we've summarized some of the candidate genes that might be relevant for marine calcifying organisms, the ones that don't have fully sequenced genomes, which is most of them. So you can see there are genes like nacrine or nacrine-like proteins. There's some sequence available for oysters, scallops, snails. Okay, this is not an inclusive list, but it's a good place to start. So we basically went into the database, looked for genes that we know are important in the calcification process in this case, and sort of compiled what's known. Okay, so if you want to measure RNA, the first thing you need to do is isolate it. And so this is a very standard technique called a phenochloroform extraction. And in this case, I'm showing you a purple sea urchin, and we've selected tube feet as the tissue that we're going to investigate. But we often do this with whole larvae that we grind up. You can do it with any tissue that you want. And you do an extraction, and in the end, you get a little tube full of your total RNA. So you get rid of DNA, protein, clean it up really nice, and have this RNA suspended in water. And from that RNA then, what you need to do is you need to make cDNA. And so you start with your mRNA template, and fully formed mRNA molecules have you see right here, this poly-A tail, and so that's an advantage because what you do is you use a sequence of T's, a primer, and an enzyme called reverse transcriptase, and you make a complementary DNA copy of your mRNA. And then it's from that DNA molecule then in your sample that you can then assay how much of a particular cDNA, which is representative of your mRNA molecule, is in your sample. So once you have this nice population then of cDNA molecules, you can use these molecules then to undergo a polymerase chain reaction, or a PCR. And so this is just to sort of remind everyone how a PCR works. And you have your DNA template, your primers, which are specific to the target that you're interested in, so the gene that you're particularly interested in amplifying. You go through a series of temperature changes, so heating and cooling, and you get a doubling of your product with each cycle. So with each cycling, you get a doubling of the product that you're particularly interested in. And the reason I'm sort of describing this is because we can use PCR in a quantitative way to measure how much mRNA or cDNA of a particular type we had to start. And so the technique is called quantitative PCR, or real-time PCR, and all it is a way to look inside your PCR tube and measure in real time how much product you're making. So it's really not that different from a standard PCR. It's just like being able to look into the tube and actually see how much product you're making. And what's the key thing is that the amount of product that you're making at any given time depends on how much template you had to start with, or how much mRNA you had in your sample to begin with. So if you compare a heat-stressed animal sample with a control sample, you would think that the heat-stressed animal would probably have more HSP mRNA signal in the sample relative to the control. And so if you ran that in a qPCR assay and you quantified, it's a fluorescent assay, so if you quantified fluorescence over time or over cycle number, you would see that samples that had a lot more HSP in them to start would pop up first, so they would come up earlier compared to control samples which would come up later. Okay, so that's a way to measure how much you had as your starting product. So that's the way quantitative PCR works. Does that make sense? Pretty good, yeah. And this is the type of data. This is just a sample of some data that you might get for heat shock protein mRNA expression. And what I'm showing you here is, in this case, this is from fish, gill HSP 70-2, which is an isoform of one of the 70 kilodalton heat shock proteins, over different controls and heat stresses. And you can see that with increasing temperature, you get this really nice increase in HSP mRNA expression. And the other thing you can see is that northern fish are in blue, southern fish are in red, and these fish are very closely related, thought to be locally adapted to thermal conditions, but are very, very similar to one another. But you can pick up differences in HSP 70-2 expression in this case with not really dramatically different treatment levels. So it's a very sensitive technique, and it works really well. Okay, so that was an example of how you might do a candidate gene approach. So using quantitative PCR to look at a specific gene that you think has a certain role under a certain set of conditions. The other thing that you can do is you can take a genome screening approach. So you can look more wholly at expression patterns of animals and look at many, many genes at one time. And this is where you often hear about microarray technology. So array technologies are involved in this sort of genome screening approach. Okay. So the how-tos of microarrays this is sort of the basic of how microarrays work in general. So you have a DNA microarray, which is a slide or a chip that's coated with spots of DNA. It's just like a microscope slide. And each spot on that chip is representative of a gene. So these probes are specific to different genes. So all these spots, in theory, each little spot is a probe for a different gene. And so what you can do then is you can take your mRNA, again isolated from, in this example, your stressed and your non-stressed samples, make it to cDNA again, but this time you make it into fluorescently labeled cDNA, and then you combine your samples, smear them over this chip, and let them hybridize onto the DNA probes. And it's a lot easier if I just show you sort of the schematic of how it works. So you have your control samples and your experimental samples. You isolate the RNA. You label the cDNA with fluorescent tags. And in this case, I'm showing you the experimental sample is labeled with a red tag. The control sample is labeled with a green tag. You competitively hybridize these samples onto the array, and then you get an image or a heat map, which is what's shown here on the right. Okay, and so this is what the data would look like. So this is an image of the whole slide, and I've just blown up a section. And so what you can see is that the spots are all different colors, and they have different intensities, and these colors then indicate how much mRNA you had in your experimental versus your control samples when you hybridize them on the chip. And so a red spot, like this one, indicates that whatever gene that probe is specific for is upregulated or turned on, or there's more of it in your experimental red labeled sample. If you see a green dot, that means your experimental sample is downregulated relative to the control, or the control sample has more of that particular transcript. And then yellow is that there's no difference. And so you get varying degrees of color, and then you can quantify the fluorescent intensity, and you get a relative measure of gene expression between a control condition and a stressed condition. Okay, so just to kind of summarize the basic transcriptomic approaches, again, you have the candidate gene approach, where you can really ask really hypothesis-driven questions. So you have a hypothesis in mind, you think a gene's important, you can test that specifically with a candidate gene approach. And then the information that you glean from that may send you on a path of looking at how this gene is regulated. If you see something really interesting, maybe the regulation is the feature that's giving you the signal. You can carry on to look at protein levels and function. So lots of things come out of looking at gene expression data. Genome screening can also be hypothesis-driven. If you have heat-check protein genes on your chip, then you can ask similar questions. But it also is good because it has a discovery component. So it may point you in the direction of some pathway or some particular suite of genes that you might not have a priori thought were important, but they may show up as giving you a strong signal when you run your array, and then that can help you to identify pathways that are key or pathways that may be important. Okay, so that was kind of the very quick tour through some of how transcriptomics work on the technical side of things. And what I want to do now is show you how you can apply transcriptomics to ocean acidification experiments. And in this case, I'm going to show you some data from the Hoffman lab on using the purple sea urchin. So we're trying to use transcriptomics to gain physiological insight. And we're using the purple sea urchin for a couple of reasons. It's a calcifier, and it also has tremendous genomic resources. So with the purple sea urchin, we have the advantage of having a fully sequenced genome, and it's largely annotated, so we know what the function or what the name and often the function of the genes that have been sequenced in the genome are. So it's a really, really powerful system because of the genomic resources available. So the types of questions that we're interested in asking are, do marine organisms have the physiological plasticity necessary to compensate and or tolerate for future climate change scenarios? More specifically, the things that ecological genomic or insights from ecological genomics can help you with are things like, in the future, will urchins display a stressed phenotype? Can we identify some signal of stress? Will they have the capacity to create and maintain functional skeletons? Do they have an enhanced sensitivity to environmental stressors? is there some alteration in the timing or the coordination of development? So we do a lot of work with larval sea urchins, and we track them through development under different ocean acidification conditions. And so will the timeline and the coordination shift? And then also to ask the question, is there going to be a shift in the energetics? If they're shifting their gene expression and having to use lots of pathways that they would normally use, will we see that show up in the energetics? So will they have to compensate in some way? Okay, so for the data I'm going to show you now, the experimental approach that we've taken in this data set is to rear the purple sea urchin larvae at 15 degrees and then expose them to one of 3 IPCC scenarios for atmospheric CO2 level. So in this case, I'll show you a control, which we had at about 380, the B1 scenario at 540, and then a higher CO2 level, thereabouts 1,000 ppm. And to simulate the ocean acidification conditions, we have a gas mixing system in the lab where we can generate air at these different CO2 levels, mix it into reactor buckets, and then deliver this CO2 equilibrated water to our larval cultures. And then we rear larvae. So this is a male sea urchin, and this is a female delivering her eggs into a beaker. We spawn the larvae, fertilize them, and then expose them to these conditions. And so here I'm showing you a blastula, which at 15 degrees is about 20 hours old, roughly. This is a late gastrula stage, and you can see here the spicules, the early stages of some of the skeletal units are beginning to form. So this is, I think, about a 45-hour old embryo. And then this is a forearm stage, and these are the data that I'll show you today. So this is a 70-hour purple sea urchin larvae with well-defined skeletal rods. So they're clearly undergoing biomineralization. Okay, so this we have a very similar design to what I showed you before. So the microarray experimental design then was to take control larvae and compare them to experimental or elevated CO2 reared larvae, isolate the RNA, fluorescently label it, competitively hybridize it, and then perform our image analysis to assess which genes are upregulated and which genes are downregulated under these conditions. Okay, so we know that the response to altered seawater chemistry is likely going to be very complex. It's going to involve several cellular and molecular mechanisms. Okay, and what we're trying to do is really to look at a large suite of genes all at one time to try to get hints at what pathways are important or what things are changing. And I'm going to spend a couple seconds talking about this particular microarray because it's not as clear-cut as the candidate gene approach and the genome screening approach, because what we've been able to do in this case is to use a combination of those two approaches. Because we have the sequenced genome of the purple urchin, what we can do is we can actually go in and say, I would like the sequence for these 20 genes that we think would be important in a certain condition. So it's like doing the same thing I described for HSPs, but we can do it for hundreds to thousands of genes at a time. And so what we go into the database, we pick genes that we think are interesting, and instead of spotting random probes onto the chip, we actually make probes for the sequences that we obtain. So in this case, we know what each of our probes, each of our spots is, because we designed the probe for a specific gene. So it's called an oligonucleotide array. It's one of sort of the newer morphs of microarray technology. And in this particular array, which was designed by another postdoc in the lab named Ann Tajam, she focused on stress-responsive pathways, so things that we think will certainly pop on in response to ocean acidification. So heat shock proteins would probably be likely, biomineralization genes would be likely. Okay, so a whole suite of genes that we think, based on sort of our a priori knowledge of how these things work in urchins, we've designed a stress-responsive chip in this case. These chips are very sensitive, and you can pack a lot of probes, so they're high density. So you could really pack in a lot of probes onto a very small space. And you can also, in this case, what I'm showing you is a four-plex array. So instead of having an entire microscope slide for 2 samples that you competitively hybridize, in this case, and we're using a four-plex array, you actually can have 8 samples and then hybridize them. So you can sort of, it's a bit more high throughput. Okay, so what Anne did is she thought really hard about what genes to choose, and this is just an example of some of the biomineralization genes that are on there. So she targeted 3 important classes of proteins that are involved in the organic matrix, or the spicular matrix proteins, many of the membrane transporters. You'll see carbonic anhydrase, and many of the isoforms are carbonic anhydrase, and Brad sort of talked about the importance of this particular enzyme. So basically we went through many of the different categories of genes and sort of did this for all of them. So what you'll see is that our candidate pathways are sort of broken up into these clusters. So we have biomineralization representatives on the chip, a number of things that are important in acid-base balance, cellular stress response. Okay, so that was our approach. And in the end, it turned out that the oligoarray targeted 1,100 candidate genes roughly, and it's called a targeted or a boutique array because we actually were able to hand build it, and so it's like a personalized, kind of like a personalized boutique is a personalized store. It's kind of the same thing for the array. Okay, and just to sort of illustrate how this chip works a little bit, I'm going to show you 2 different data sets. So the first one I'm going to show you is when we exposed control larvae just to a simple heat stress. So we asked the question, do these larvae demonstrate the classic heat shock response? And can we show that with the chip experimentally? And I've just selected a few of the genes that are, particularly interesting. So what we see is with heat stress, as you would expect, the volume is up, or genes are upregulated in response to heat stress. And I'm showing you molecular chaperones. So these are heat shock proteins that we know play a role in animals in response to dealing with thermal stress. There's also some ubiquitin genes, which are involved in degrading damaged proteins that may be damaged because of the heat stress. A number of other genes are turned down. And so in this case, biomineralization genes are turned down. And this is what you would expect for the classic heat shock response. So animals turn up heat shock proteins and things that are directly involved with dealing with heat stress, and everything else is turned down. So they preferentially synthesize proteins that are involved in dealing with the stressor. So this is a demonstration of the classic heat shock stress response. Okay, so it looks like our chip is working. It's quite sensitive. And now what I'm going to show you is what happens to purple sea urchin gene expression when they're reared under 380, or sort of a control level of CO2 and an elevated level of CO2. And we don't really have a lot of predictions for what we think might happen. There are a couple. In Brad Seibel's talk, he hinted yesterday a little bit about we know that oxygen or CO2, low oxygen or elevated CO2 may trigger a metabolic suppression. Okay, so that's one prediction maybe that we might have that he hinted at. And so what I'm showing you here is in response to ocean acidification, then in purple urchins, biomineralization genes are actually turned down. So a bunch of the genes that are involved in, or that are found in the primary mesenchyme specific proteins are all turned down. And I sort of gave away the punchline on this one by mentioning what I just mentioned, but in response to ocean acidification, metabolic genes are also turned down. So this is consistent then with some of the mechanisms that Brad was describing in his talk yesterday. So a number of enzymes involved in the TCA cycle, some involved in electron transport. So suggesting that the metabolism of these animals in response to elevated CO2 is in fact turned down. And then some of the cellular stress and defense genes are also turned down. So you see an HSP popping up here. Some of the xenobiotic and toxicant defense genes are turned down. So in general, and this is more, I'm only highlighting a few of the pathways, but in general, we saw an overall downregulation of genes in general in response to elevated CO2. So just to sort of summarize, it looks like purple urchins in this case are downregulating many of their genes. So almost hunkering down and sort of perhaps waiting for things to get better. And so this brings up sort of another point that came up yesterday, is there a critical threshold of CO2 that we can address using genomics, or are these data indicating that there's something really happening at 540? And what I can tell you is that in purple urchins, if you look at what happens to them at 970 ppm CO2, everything is still down. So it looks like for purple urchins with elevated CO2, everything is being turned down. It's very, very consistent. I think of the 1,100 genes, only 1 or 2 were significantly upregulated. So it's a very obvious, very clear picture in purple urchins anyway. But the next point I wanted to make is that you can use these chips to look at other species. And so we don't have fully sequenced genomes for other urchin species, but what you can often do is get lucky that enough sequences will be similar enough in these closely related animals that you can use a purple array, a purple urchin designed array, to test the response in another species. So a heterologous hybridization. And what we've done is we've done this experiment with using the purple array with Lidocynus pictus. So reared in the same way that I described for the purple urchin. And I'm not going to show you the data, but what I'll tell you is that the story is very, very different. And so if you're thinking about, generalizing these responses in the 2 species that we've studied, and we're already seeing really different patterns of expression. And so Lidocynus pictus reared at 540 actually upregulates a lot of genes, including biomineralization genes. And then at 970, they begin to turn things down. So the response between these 2 species is quite different. But it's something we're actually able to detect using these sorts of technologies. Another thing that we're interested in understanding is that is there evidence for cost growing up in a high CO2 world? So if we raise larvae at 15 degrees and we give them a thermal challenge and ask the question, do they do just as well as larvae reared under control CO2 levels? What this data actually comes from Moose O'Donnell when he was a postdoc in the lab. He showed that there's a reduction in the expression of molecular chaperones in response to a heat stress if you grow up in a high CO2 world. And so data like this are really important because what they suggest is that if you have a lower level of mRNA for defense genes, in this case HSPs, that may lead to a reduction in your thermal tolerance. So you can begin to ask questions about synergistic stressors and how that might impact an animal's performance in the context of future climate change scenarios. Okay. So I really have gone really quickly, so I only have a few slides left. But what I want to end on is just to sort of give you a hint at some of the next generation sequencing technologies that are going to be coming online that we think have relevant applications for genomics experiments in the context of ocean acidification. Okay, so I tried to kind of did a not full summary of some of the genomic resources that are available for calcifying organisms that might be particularly interesting. So we know that urchins, S. purpuratus, is fully sequenced. Oysters, the genome is in progress and I think is relatively close. My impression from talking to people is that the holdup is in the assembly, so they have the sequence done, now they're trying to assemble it and organize it. Abalone, scallop, and mussels have a select number of genes. I think there's one species of coccolithophores that now has a genome. I don't think it's been released yet, but I think it's going to be coming soon. Corals don't have too much in the way of sequence, but again, people are trying to work on this, so in the next couple years, hopefully there will be some coral genomic resources. And then pteropods have virtually little to no sequence available for them as of yet. And so the reason I'm talking about next generation sequencing is that this is these sequencing technologies are high throughput sequencing technologies, and many new ones are emerging, and the literature is evolving very, very quickly. So I probably worked on one of these slides a couple months ago, and the table that I'm going to show you is probably going to be a little bit out to date already. So there are many companies working on the chemistries, and the chemistries vary, so it's a very competitive and quickly evolving field. And Gretchen calls it the Wild West in molecular biology, which I think is very, very accurate because it really is the Wild West. So the field's moving quickly. We're not sure which direction or which particular technology is going to come out on top. And there are 2 relevant applications that I just sort of want to point you to. And so why you might want to use next-generation sequencing technology is to obtain sequence for species with no genomic resources, and I think I've demonstrated that if you have tremendous genomic resources for a species, you have the ability to really do some very targeted experiments. The other application that is going to be kind of neat is to actually use the sequencing technologies to count the abundance of transcripts in a sample. So instead of doing qPCR or microarrays, it may very well be that you can actually take your RNA sample and sequence that, and then from the sequence then you can count the transcript type that you're interested in. This is going to be a ways away, and it's very, very expensive. So to do an experiment like that on 64 samples that you generate would probably be astronomically expensive, and it's about 10 times at least the cost of using some of the other approaches that I've described, which are already really expensive. But it's pretty exciting because every 6 months, the costs of some of these next-generation sequencing technologies seem to be cut in half. So it's all happening really, really quickly. And some of the terminology, because there's all sorts of terminology associated with next-generation sequencing. You'll see the sequencing by synthesis method, which refers to Solexa and Illumina technologies. You'll also hear the word pyrosequencing, which is the sort of new word for 454 sequencing. And again, this technology is advancing very, very rapidly, and the bioinformatics are going to have to sort of keep up. So we started a 454 sequencing project. We got a little bit of sequence back and realized that, wow, the bioinformatics are going to be really tricky. And so that field is also going to have to evolve pretty quickly in order to make this something that we can use very effectively. And I put this in here. I got it from actually Anderson Mayfield, who's also here at the course. And just to sort of have this in your reference, just to kind of note that the chemistry is different between these different types of sequencings. The read lengths are also very different, and the outputs that you get vary between platforms. And so you get these different read lengths, and so depending on what type of analysis you're interested in, a 35 base pair chunk of sequence may be good for you, or you may need something much longer. So that's sort of one thing to consider. And then the amount overall output varies. And then the actual amount that you really get when you send off a sample to a sequencing lab doesn't usually follow the manufacturer's sort of proposed how much you actually get. So it's coming, but it still has a long way to go. Just to kind of show you some interesting data, and something that Gretchen's working on that I think is really neat, is she went to the Antarctic and collected some pteropods and exposed them to all sorts of different stressors and brought those samples back. And I've developed a normalized cDNA pool from all these combined samples to try to get a representative pool that has lots of different transcripts upregulated under lots of different stressful conditions, and then sent this off for 454 sequencing to try to gain a lot of sequence for a species that doesn't really have any. And so we're at the very preliminary stages of looking at these data, but overall, on a reading of a half a plate, I was able to get about 4,500 sequences, and this just kind of shows different species that they align with, and then the number of sequences that I gained with different lengths. And so in theory, I was supposed to get lots of sequences of 400 and 500 base pair lengths, but most of my sequences were a lot shorter than that, even after assembly. So the technology has a little ways to go. Okay, and with that, sorry I kind of went so fast, but I guess that's good. Well, if you guys want to have some questions and just talk about sort of genomic approaches, I would be happy to, and I'm around all of today. I'm going to leave tomorrow morning, and Anderson is also here, and he's a really good coral molecular biologist. So for those of you that are interested in coral molecular techniques, he'd be a great person to talk to. So thank you very much. Hi. Sure.
I'm not a geneticist and I work with corals, so I don't deal with organs normally. But would it matter, I mean, RNA is obviously expressed on an organ-specific lane, so you would not take a QPP to look at calcification.
Right. Yeah, so you would pick, one of the things you might do is you would have to determine what tissue or where that particular gene is being expressed. So you would want to know what tissue is most sensitive. So we look at whole larvae just because they're so small. We grind up the whole thing because we need thousands of them to actually get enough sample to do analysis on. In fish, we often look at the gill because the gill is in direct contact with the water, so it's very sensitive to thermal stress, for example. So, yeah, you definitely have to pick the right tissue. 2 feet would probably be not a good calcification organ choice. Yeah.
I do microarray work in a different way, where I have one variance of one gene and looking at community composition.
Uh-huh.
And in that case, we have to be very concerned about the thermodynamics of the array, and making sure that differences in sequence don't affect the binding capacity of the array. So my question is, in the competitive arrays that you use, you have a sequence that you derive from the Urchin database, and then your treatments, you're going to have used. Is it a very highly conserved region of the DNA that you use for the array, or if you perturb your system, do you anticipate that there would be some mutations in the DNA that would change the thermodynamics of its binding to the array? Do you understand my question?
Yeah. So I don't think you ever fully know, because certainly most of the probes that you design should be very, very specific, and the chances of you having non-specific binding are low because you know all the sequences really well. But certainly that can happen. So, but I wouldn't expect it to be a huge problem. Does that make sense?
So you use one probe at a time, or you use a mixture of probes?
So your chip has a whole lot of probes on it. And actually each gene, so this actually kind of helps, each gene that we have a probe representative of on the chip has 3 different types of probes for that gene. And so if I took an HSP sequence, I would actually develop 3 different probe types for that gene, different sequences. And so you should expect to see if that probe is working. You would see the same thing on each of those 3 spots. And so that's actually a quality control thing that you can look for. Yeah. But when you hybridize your samples to the chip, it's a mixture of all the RNAs in your experimental sample and all the RNAs in your control sample. They all have label, and then they bind to the complementary sequences of the probe that they match with.
But that's the optimum temperature might be different for these genes then.
I don't think, because their probe lengths are short, and so we do the hybridizations at a constant temperature. And it's all just based on the thermodynamics of the actual complementary sequence binding. It doesn't really have a lot to do with the particular thermodynamics of the gene itself.
The different lengths of sequence would have different annealing temperatures, yeah?
Yeah, but I think we do them at 65. So, yeah. Hi.
Could you say something in the context of adaptation, acclimation, and survival of organisms in terms of this technique? Like if a gene is expressed or not expressed, it doesn't mean that the organism won't survive.
Yeah, so when we do these experiments, generally what we do is we look at look at gene expression, look for patterns, look for things that are important, and we usually combine them with some other measures of animal performance. So we look for thermal sensitivity, so we might do thermal tolerance. And so it's really hard to make the link between mRNA expression and will it even actually reflect the protein pool? So sometimes you get a lot of transcripts, are they actually translated into the actual functional proteins or the functional units? And so that's always one of the things that you have to follow up with other sort of functional genomics types of approaches to actually understand if the gene that you're measuring that's changing is at all important for the animal in an acclimation context, in an adaptive context.
As someone is not familiar with this technique, I noticed on some of the slides you had, it looked like your error bars were as large as your data bars. Is that a concern in interpretation?
On the genomic data?
Regulation. You were showing up and down regulation. You had error bars that looked fairly large.
Yeah, so the error bars are fairly large, and if you actually look at the scale, the changes are very, very subtle. And so the statistical approaches that you use for microarrays are really it's tricky And we actually work with a microarray statistician to make sure that we actually do this correctly, because the other thing that I didn't mention that's very important is that microarrays usually, if you make them from a cDNA library, sort of the way they've been done in the past, they have a whole variety of genes on them. You don't necessarily know what they are, but there's a whole bunch of genes on there that don't do anything at all, so that don't change. And so you can use that background no change as a way to sort of control, as a kind of a control expression almost. But what you have to consider with a boutique array or this targeted array is that we expect, 60% more than that of these genes to change with our stressors. And so that changes dramatically the statistics that you can use. And so we actually have a statistician that helps us design our hybridizations appropriately, because there's all sorts of technical things that I didn't talk about, like dye swaps and making sure that your green label works just as well as your red label, because if you label all your samples experimental control in the same way and you get differential binding because of the dye, that will influence your results. So there's a lot of complexity that I didn't talk about, but the statistician helps us make sure that we actually are picking up changes that are significant. So all the ones that I showed you were statistically significant deviations from control.
So do you think that the 4x4 sequencing method is more robust? So the interpretation of those results, so to speak, about the quantitative changes in these?
I think it's going to be a long time before it's going to take a long time before they are as sensitive as qPCR. So qPCR is very, very sensitive. You can detect 1 or 2 transcript abundance differences in samples. I mean, it's very, very good. The oligo array is also very good. Maybe not quite as good as qPCR, but pretty close. In theory, the sequencing should be able to detect every single thing, but there's always sequencing errors.